All right, let's get right into it. Now, you probably remember this from high school biology, right? DNA makes RNA and RNA makes protein. The central dogma. Simple. But what if I told you that one of those molecules, RNA, is hiding a secret? That it's not just some bit player. It's actually the secret superhero of the story.
Yeah, the textbook story is super straightforward. DNA is like the master blueprint, right? It's locked away, safe in the nucleus. And when the cell needs to build something, it makes a quick, disposable copy. That's our messenger RNA. That little messenger zips out to the cell's factory, the ribosome, delivers its instructions, and poof, you get a protein. It's this neat, tidy, one-way street. DNA to RNA to protein.
But here's the thing. What if that tidy story is just, well, wrong? Or at least not the whole story? What if that messenger wasn't just delivering the mail? What if it was also drawing the blueprints, laying the foundation, and running the whole construction site? That's the rabbit hole we're going down today.
So let's start by totally dismantling that old picture of RNA. We're going to see why calling it just a messenger is a massive understatement.
So on one hand, you have the myth. RNA is just a simple, throwaway copy of DNA's instructions. But the reality, oh man, the reality is so much cooler. Scientists have found it's this incredibly dynamic molecule. It's a structural architect, a chemical catalyst, a genetic light switch, and for a whole bunch of viruses, it's the entire genetic blueprint. I mean, how can one molecule do all of that? How does it wear so many different hats?
Well, the secret, the key to all RNA's superpowers, it's not just in its sequence, it's in its amazing ability to fold.
You really have to think of it like molecular origami. Imagine this long, flimsy ribbon. That's the single strand of RNA. On its own, it's not much, but it can fold back on itself, twisting and turning into these really complex, stable 3D shapes. And it's those shapes, that final origami creation, that give RNA all its incredible functions.
And this is where it all starts, the 2D blueprint. You can see how the RNA strand folds back and parts of it pair up, creating these little ladder-like sections we call stems. And then the parts that don't pair up, they form these hairpin loops. These are the basic folds, the first simple creases in our piece of molecular origami. Okay, but a flat blueprint isn't a machine, right? So how do we get from this simple 2D plan to a full-blown working 3D machine? That's where things get really clever. Even way back in 1966, people were starting to get suspicious. Francis Crick, one of the guys who discovered DNA's structure, he looked at this thing called transfer RNA, or tRNA, and said, "It looks like nature is trying to make RNA do the job of a protein." He saw that it wasn't just a string of letters, it looked like a complex tool. And boy, was he right.
And this is exactly how that happens. You see that 2D cloverleaf shape of tRNA? It doesn't stay flat. Those different stems, those little helical ladders, they actually stack right on top of each other, almost like you're snapping Lego bricks together. This process, called coaxial stacking, transforms the whole thing from a floppy cloverleaf into a rigid L-shaped structure that's ready to go to work.
But just stacking isn't enough to hold these really complex 3D shapes together. For that, nature has this entire toolkit of special, clever folding patterns called tertiary motifs. Think of these as the advanced origami techniques, the fancy folds that lock the final structure in place.
So let's look at a couple of these. First up, the pseudo-knot. This is basically like tying a knot in the molecule. You see how a loop from one part of the chain reaches way over and binds with a totally different section? That locks two regions together and creates this super stable core for the structure.
And then you've got this for precision work, the A minor motif. This is just incredible. A single A base from one part of the chain slots perfectly into a groove on a helix somewhere else. It's like a key sliding into a lock, creating these really specific, really stable connections across long distances.
And finally, if you're going to build real architecture, you need corners, right? You need sharp angles. That is the kink turn's job. It acts like a specialized hinge, putting a sudden sharp bend into the RNA backbone. It's this whole toolkit, the knots, the locks, the hinges, all working together that lets RNA build things just as complex as any protein.
All right, so we've seen how RNA can build these amazing shapes. Now let's talk about what these shapes actually let it do in the cell. And the job list is just mind-blowing. Its shape allows it to be a template for finishing off our chromosomes. It acts as a scaffold, like a molecular workbench, for building other huge protein machines. Its structure forms the literal engine of the ribosome, the machine that builds every single protein in your body. It can be a tiny sensor that changes shape to turn genes on and off. And for a ton of viruses, like the flu or COVID-19, RNA is the genome. It really is biology's ultimate multitasker.
But out of all those incredible jobs, one of them, acting as the engine of the ribosome, was a clue to a secret that, once discovered, would completely shatter our understanding of how life works.
See, for decades, there was this one central rule in biology that was considered absolute. Enzymes, the catalysts that speed up all the chemical reactions that keep you alive, could only be made of protein. That was it. RNA carried the information, proteins did the work. End of story. It was dogma.
And then, the dogma was broken. In the late 70s, a scientist named Thomas Seck was studying a simple little organism. He was trying to figure out which protein enzyme was responsible for processing its RNA, but he couldn't find it. Because, to his total shock, the RNA was splicing itself, cutting out a piece of its own sequence and stitching the ends back together with absolutely no proteins around. It took years of super careful experiments to prove it wasn't some kind of contamination, but in 1982, they did. A year later, another lab, Sidney Altman's, confirmed it for a different RNA molecule. This was a biological earthquake. And in 1989, Seck and Altman rightly shared the Nobel Prize for it.
And this right here, this is the smoking gun. This image is the hard proof. It's basically a picture taken with radioactivity, and it shows, without a doubt, that pure RNA all by itself in a test tube could perform chemistry. It could cut and paste its own code. The central rule was officially dead.
And this massive discovery gave us a whole new word for our vocabulary, the ribozyme. It's a mashup of ribonucleic acid and enzyme. And for the first time, we had to accept that RNA could be both the message and the machine.
The discovery of ribozymes didn't just rewrite the textbooks on the modern cell. It blew the doors wide open on a much bigger question: how life itself got started on Earth.
Because it offered this beautiful, elegant solution to biology's ultimate chicken or egg problem. Which came first? DNA, the genetic blueprint, or proteins, the machines that do all the work? You need DNA to make proteins, but you need proteins to replicate DNA. So, which one was first? The RNA world hypothesis says neither. It suggests that on early Earth, there was a time when RNA did everything. It was both the genetic code, like DNA, and the functional enzyme, like proteins. Life could have kicked off with simple RNA molecules that could catalyze their own replication.
So when you put it all together, you realize something profound. RNA is not just a supporting actor. It's an architect, it's a machine, it's a catalyst. It's basically a living fossil from the dawn of time that is still running critical parts of our cells today. And that leaves us with one last mind-bending question. Could the entire epic story of life, the one we all thought was written in the language of DNA, have actually begun with a single, humble, and unbelievably versatile strand of RNA